So we're going to talk to you tonight about a lot of things that have to do with this virus that's named SARS CoV-2. We're going to give you some information about the virus, some information about how it spreads, and also the role of the immune system in vaccination. But we'll also have time at the end of the presentation to answer as many questions as you may have, because as we're putting together this presentation, we started to think of frequently asked questions, and we realized that there are a lot of them, and we can probably spend hours just compiling so many. So we're going to leave it up to the audience to come up with any of the questions that we might not address in the talk today. So I want to start with just talking about what viruses are. So everything that has to do with this pandemic is based upon a particular type of virus. And viruses have been around since time immemorial. These are non-living entities that rely on another organism, a host, for survival. And these viruses are very specific in what they can infect. So when we have a virus such as what's called a bacteriophage, this type of virus can only infect bacteria. A tobacco mosaic virus can only infect a tobacco plant. And then there are specific viruses that can only infect humans. They're very simple in structure. It's really amazing what they can do based upon their simplicity. Really, all they are is a collection of proteins. And you can see here in the adenovirus. So the adenovirus is a type of virus that can cause certain types of colds. It's also used in delivering vaccines. You can see it's surrounded by these proteins in the capsid. And then inside of these viruses, you have the genetic information, which is either DNA or RNA. They're viruses that can infect every living organism. We're going to focus on coronaviruses tonight. Coronaviruses have what's called an RNA genome, and there are a number of different types of coronaviruses that are present in the world. Now, we're hopefully on the tail end of this COVID-19 pandemic. This is not the first pandemic when it comes to a coronavirus. And in the last 2 decades, there have been 2 other ones. And you may remember some of these. I remember both of them pretty well. The first one occurred in 2002. This was the what was called the SARS outbreak. This started in China. And then in 2012, there was the MERS outbreak. And then starting in December 2019, we had the emergence of the current virus. This is an older slide. It has it as 2019 nCoV. It is now officially, the name of the virus is officially SARS CoV-2. These first 2 outbreaks did not spread to nearly the global level that the current outbreak has. These numbers are from about a week and a half to 2 weeks ago, so there may be some slight updates, but you can see a lot more countries have been affected by this current outbreak, and more than 110 million people have been confirmed infected with SARS CoV-2 compared to just in the thousands of the 2 previous outbreaks. The number of deaths is dramatically higher. We're probably over 2.5 million deaths at this point, compared to in the hundreds with these previous 2 outbreaks. However, the death rate is actually lower for SARS CoV-2. The MERS outbreak was a very scary time because the death rate was actually about 35%. But what we see with this current outbreak with SARS CoV-2 is that the spread is much more dramatic. And I'll talk about a couple reasons for why that is in a few minutes. Now, all of these coronaviruses have what are called hosts. And all of them have what are referred to as reservoir hosts and intermediate hosts. So this is a list of most of the known coronaviruses in the world. The ones that I just spoke about are these in red, SARS CoV-2. So that was the 2002 outbreak, the MERS outbreak, and then the current ones, the SARS-CoV up here and the SARS CoV-2. And these ones that say HCOV, these are coronaviruses that cause the common cold. So these are prevalent in our world as well. All of these originated in either bats or rodents. So we can trace all of these coronaviruses initially back to either bats or rodents. In order for the virus to infect humans, it needs to go through an intermediate host. And these different viruses have different intermediate hosts. The original SARS outbreak in 2002 was through civet cats. The MERS outbreak, this was in Saudi Arabia, was tracked down to dromedary camels. And it is thought that the current outbreak of SARS CoV-2 occurred through pangolins, which is an organism that is in the eastern part of the world. It's not completely sure that this is how what the intermediate host is, but there's some good data for that. The interesting thing about these types of viruses is humans become infected when exposed to these intermediate hosts, but there are mutations that need to occur in order for these viruses to ultimately spread person to person. And we are at that point now with SARS-CoV-2. So what does this virus look like? Down here on the bottom left is a representation of the SARS-CoV-2 virus. And it is pretty elegantly simple. We have a membrane, which is the circle right here. And inside the membrane is the RNA, which contains all the genetic information for the virus. And sticking out of the membrane are these spike proteins, which you've probably heard about by now, because one, they're very important for infection, and two, this is what is being focused on when it comes to the variants, which I'll speak about in a bit. If we were to look at this RNA genome here, so this is the blueprint for everything that the virus does. It is shown up here in a linear fashion. There are about 30,000 base pairs in the coronavirus genome. To give you kind of an idea of how that compares to something like a human cell, every human cell contains 3 billion base pairs. So this virus has a genome that is 10,000 times smaller, yet it is still able to kill, millions and millions of people, or at least more than, we're in the multiple millions of people worldwide. So there are a lot of different things that are important in this genome, but I'm only going to point out two that are relevant for what I'm going to speak about tonight. The first one is this gene that says RNA-dependent RNA polymerase. So this gene right here, this codes for a protein that allows the virus to copy its genome. So every time the virus infects the cell, its target cell, it needs to make more copies of the virus. And it does that using this protein here, the RNA-dependent RNA polymerase. And then this other gene here, which is the spike protein gene. So this gene codes for the proteins that stick out of the virus that allow the virus to essentially attach to human cells. And this is what results in viral infection. So how do these viruses actually infect human cells? So this is another graphical representation of the virus. So we have our membrane, we have our RNA, and then we have these spike proteins sticking out, these ones that are in pink here. These spike proteins, as is the case with all viruses, have to interact with another protein on the outside of the cell. When it comes to the SARS-CoV-2 virus, these bind to what's called the ACE2 receptor. And your bronchial epithelial cells, as well as other cells in your respiratory tract, have these ACE2 receptors that stick out of the cells. So what happens here is that when you inhale the virus, because the vast majority of, and to my knowledge, all of the documented cases of transmission are through respiratory events. When you inhale this virus, it is going to stick to these receptors that are on the outside of the cells in your respiratory tract or in your lungs. The virus is going to essentially poke through the membrane of the cell, and it's going to get released into the cell. The virus breaks apart, and the RNA genome is now inside the cell. At this point, this protein, this RNA-dependent RNA polymerase, is going to make lots of copies of the RNA. And then it's also going to make these proteins, the spike protein, the capsid proteins, and it's going to package up that RNA, and it's going to release those viruses to go infect other surrounding cells. And that is essentially the life cycle of the virus. That is all it does. A virus is nothing, all it does is infect cells and make more viruses. And quite frankly, it's very good at it. So this is the case with virtually all viruses, but not all viruses cause pandemics like SARS-CoV-2. So one important question is, what makes SARS-CoV-2 different than other viruses? And there's a number of reasons. One of them is that this is a new virus. And while new viruses do come about, they're not that common, at least not ones that cause significant infection. The human immune systems are naive. They have not seen the virus before. And Dr. Moran will talk more about how the human immune system responds to a new virus or a virus in general. But when there's a new virus and the immune system has not been primed to see it, the immune system has trouble responding. Another reason that this seems to be different has to do with those ACE2 receptors. So these ACE2 receptors, while they are expressed in the bronchial epithelium and in the epithelial cells of the respiratory tract, they're also expressed in a number of other parts of the body. For example, the ileum, which is part of the small intestine. We know that the virus can be shed through the digestive tract. And in fact, one of the ways to predict outbreaks of the virus is to look at wastewater. We know also that there are a lot of examples of COVID-19 having significant effects on the brain. The brain expresses these receptors along with other tissues in the body as well. So this virus can infect cells other than just those in the respiratory tract. And that's why we see such a wide array of symptoms sometimes. It seems to be also that this virus has the ability to stick to cells much greater. And this will be very relevant when Dr. Alvey talks about the way that you can measure and predict viral spread. But if a cell has an ability to stick much greater to its receptor, what that means is you don't need as many viral proteins in order to get sick. And there is a number of viral particles to get sick. There is a minimum number of viral particles for every virus that you need in order to get a productive infection. This virus seems to have a relatively low number because of that ability for the receptor to bind to the spike protein. And perhaps one of the most important ones is that this virus really does have the ability to be spread while asymptomatic. So on this graph here, on the x-axis, the 0 time point is the day in which symptoms arise after infection. The green line right here represents the amount of viral genome that can be measured in the body. And this is typically done through, as people probably know now, a PCR test. If someone is sorry, my mouse is going a little crazy there. If someone gets infected at day -6, for example, 6 days before symptoms, and we know that symptoms can take up to 14 days to show. But here, if someone gets infected 6 days before symptoms, they have a very high viral load in their body before symptoms even occur. And this has become a really big issue with COVID-19 because there appears to be a large amount of asymptomatic spread. Now, we're doing a good job in getting towards vaccinations, which is going to be a very important and probably the most important key in defeating this virus. But as everyone probably knows, there is the issue of new variants. So we should probably define variant. Sometimes the term strain and variant is used interchangeably. We call these variants because we don't know how distinct they are from the other variants. So a variant is due to simply a mutation. So these variants have mutations compared to what we would consider the original strain, the strain that came into, and there is evidence now that the original strain came into the United States perhaps as far back as November of 2019. But this is a picture from the New York Times. They have a very nice page where you can look at the differences of all the different variants that have been sequenced in SARS CoV-2. And this represents the genome. So this is the beginning of the genome right here. And if you circle around, that's 30,000 letters, 30,000 base pairs. And each of these dots represents one mutation. So how do these mutations occur? Well, the key is this is actually a normal process in viruses. So when people start freaking out about variants, one thing you have to remember is viruses make mistakes all the time. And it's no different for SARS CoV-2. Every time the virus infects the cell, this RNA-dependent RNA polymerase is going to copy its RNA genome. It makes mistakes. It makes a good number of mistakes. Most of these mistakes will actually be bad for the virus. Some of them will be beneficial. And the mistakes that we're mostly focused on with these variants are the ones that occur in the part of the genome that code for the spike protein. Because when there is a change in the spike protein, what that does is it can change a couple things. It can change how the spike protein interacts with the receptor, but it can also change how your immune system recognizes the virus. And Dr. Moran will talk about how the immune system recognizes the virus. The main issue with the spike proteins is it appears to potentially have an effect on how antibodies can bind to the virus. Now, one thing I do want to discuss very quickly about these variants, in the media, you do hear the terms the UK variant, the Brazil variant, the South African variant. I don't particularly care for these names, because the reality is, even though the B117 virus was first identified in the United Kingdom, it is very prevalent in the United States, and it doesn't mean that it necessarily came from the United Kingdom. Because there has been so much infection with this virus, there are ample opportunities for the virus to replicate. And these different variants are showing up independent in different countries. The key is to get the rate of infection dramatically lower to decrease the opportunity of the virus to make these mistakes. So I just want to point out a couple things with the variants before I hand this off to our next presenter. This picture here is the B117 variant. Sometimes it's referred to as the UK variant. Again, most of the variants that we're concerned about have mutations in the gene for the spike protein. And we can actually break variants down into three groups. The first are variants of interest. These are variants that have some evidence that they might affect the ability of the virus to bind to the receptor. They might affect the ability of the immune system to respond, but there's not clear evidence of that. And in fact, if you go to the CDC website, there are lots of variants of interest. The current variants of concern are these five right here: B117, B1351, P1, 457, and 429. Variants of concern have documented evidence that there is a change in either the ability of the virus to bind, the level of morbidity and mortality, the ability of antibodies to recognize the virus, but there isn't enough data to suggest that these are variants of high consequence, which we have none of yet. Variants of high consequence are variants, and luckily there are none of these yet, are variants that have documented data that there are remediation measures that are not working at an effective rate, something like vaccinations, for example. Now, some of these variants, specifically 351 and P1, do show a decreased ability of antibodies to neutralize the virus. However, the recent data that has been released, and there's data coming out virtually every day, all of the recent data suggests that all three approved vaccines in the United States, Moderna, Pfizer, and J&J, do protect against severe COVID and death. It is not 100% protection against infection, though the new data suggested there's a very high protection against infection, but these vaccines protect somewhere in the order of 99 to 100% when it comes to severe COVID and death, which is something that we really want. So these variants play a really important role in the spread of this particular virus. And I'm going to hand this off to Dr. Alvey, and she's going to talk more about how we can model that type of spread.
All right. So what we want to do is take all of this information that we have about the virus itself, how it affects the body. So transmission rate, when do symptoms appear, how fast do people recover, these types of things. And then also real life data, which includes, how many people have been hospitalized, how many cases do we have total, are people quarantining, what other safety measures are they using? And we want to combine all of these things and hopefully create a math model that will allow us to kind of forecast what might happen in the future with the burden of the disease in general, and to determine control measures that might make the largest impact. And so I put on here just a very basic example, and I'll show you a more complicated one in just a little bit of what a math model might look like. So I'm starting with three categories of people. Those are these boxes here. The S, these are my susceptible or my healthy people. My I, these are infected people, and R are recovered. And basically, we're going to draw arrows to describe how people might move from one category to the next. So people can be born, people can die, from, whether or not they're healthy, infected, or recovered. This is just a natural part of life. So we're not talking about dying from a disease or anything at this point. We'll talk more about that later. Well, people can get sick, of course, that's this beta term, and people can recover. So this is a very general structure, and we're going to take that idea and expand it and see what we can do mathematically. So before we do that, I said I wanted to look at some of these main pieces of information, all of this data that we have coming through. And so on this slide, I have on the left side here, just in general, how many cases did we have for the whole of year 2020? So this is in 2020, pretty much worldwide. How many cases do we have? These are the weekly reported cases. And on the right, this is just for New York City. So the number of cases up here, these are sorted by age, hospitalizations, and deaths. And so I want to take this information and I want to use it to create hopefully an accurate math model. So to give you an example, this is just one paper that was published last year using data from China. And so what we see are, by date from January to April, this was 2020, and how many people were either hospitalized, so those are the blue data points, or the recovered individuals, those are the red data points. So all of these dotted, these points right here, all of these are actual data that we have from this place in China. And then the solid curves, these are the ones that we're creating from our mathematical model. And we can see it really matches the data pretty closely. So at least we know that mathematical models can give us some valuable information and can closely match what's actually happening in the world. And in fact, the authors of this paper were not very happy with this because they thought that the blue curve was actually overshooting the data points too much. So they actually played around with it a little bit more to get a closer fit. But you can see it might be helpful, right, to estimate the data points and then see what happens more long-term from there. So let's look at a more general model. And this model was actually created last summer. So it's actually still in preprint in the archives. And this uses the New York City data. So this big picture kind of explains what we want to consider. And we're talking about our healthy people who are becoming exposed at some point in time. And these exposed people, we're not talking about, we have our 6 feet bubbles and I stepped into someone's 6-foot bubble and, oh, maybe I'm exposed. We're talking about these people are going to get COVID, 100% going to get COVID. They just haven't quite gotten there yet. They were exposed to it. They're going to develop the actual virus and be able to infect other people. They're just not at that infectious stage yet. So they're really considered latent individuals. But from there, and this is really the important thing with COVID, that we can have asymptomatic people or symptomatic people. And so we really want to focus on what's going on with these asymptomatic people. But really our major concern is all those people who do have symptoms, a lot of them are, the more severe cases, they're going to end up in the hospital potentially. Some of these people are going to die, and we want to really limit the number of people who end up in these categories over here. So this is our ultimate goal. So this paper was created before the vaccine came out. So we have no information about a vaccine, but we know that we're social distancing, we're using masks, we're quarantining. What if we don't want to quarantine anymore? What could we say? What could we do with this model to predict what might happen if we all just restart our lives and go back out into the real world and stop quarantining altogether? So if we don't have a vaccine, what would be the goal with this? Well, hopefully we'd have fewer infections over the whole pandemic. Hopefully we would be lowering the death rate among the elderly group, which was the group that was really most affected by this pandemic in general, but lower death rates across the whole population. That would be nice. And then really when this paper was written, it was all about, all these businesses were shutting down. We had some economic issues, right? We want to get businesses back up and running, but we don't want to risk everyone's health in that process. So we want to take all of these things into account. And so what these authors did is they started a staggered release policy. Basically, this just means that instead of saying, no one has to quarantine anymore. Maybe they'll just let some people stop quarantining, and then eventually we'll ease more people back out into the real world. So this graph explains one possible strategy. So let me explain what's going on here. In this left column here, everyone is quarantining. At least 80% of people are quarantining, and that's because we have essential workers and that kind of thing who are out in the real world. And then in the second period here, let's release the young people. Let's send them back to school or to work. And I think this is kind of funny, but they define young as below the age of 35. So this is a big group. This is a lot of people right here. Then what do? Well, let's release the middle-aged population. So they consider this between 35 and 65. Let's let them go back to work, go out into their real world. And then after a certain point of time, we'll let everyone stop quarantining. And we want to see if this can help. You know, instead of just letting everyone free, let's just see if this makes any difference in a time when the vaccine wasn't really around yet. So what do these results look like? What we can see here, so time at the bottom in days, and they're talking about changing things after about 70 days. I'll talk about what these little points mean in a second. And we have the number of symptomatic infections on the y-axis. So we're talking about the symptomatic infections because we're really concerned about, you know, really lowering the hospitalizations and the death that are caused by those symptomatic infections. So that's what we're going to focus on. And I just want to point out this big giant red curve here. That's what would happen if no one quarantined at all, ever. So we can see we have a huge spike in symptomatic infections, not really a good thing. And it also goes over this level that they consider the healthcare capacity. We're not able to treat everyone. If they get sick and end up in the hospital, we're full. The hospitals are full. We can't do anything to help these people. So obviously not a good situation. That's why we're trying to implement these control measures. So this strategy is to release everyone at day 170, which is this triangle here. And the only reason there is a circular point here is because in the next example that I'm going to show you, between the star and the circle, everyone's quarantining there too. So everyone's quarantining from the star to the triangle. And you know what happens? Well, a lot of people are going to get sick. They all go back out into the real world. Unfortunately, we still go above the healthcare capacity. So that's not going to be very good in terms of hospitalizations and probably death as well. Not a good option. But if we release just that young age group first, we see a different story. So again, just for comparison, this red curve here is what would happen without any quarantining, without any interventions at all. In this first period between the star and the point here, the circle, everyone's quarantining. Between the circle and the triangle, the young age group is let go. They can go back to school or to work. And then after the triangle, everyone else is released from quarantine as well. And so you can see a much different story here. So first of all, this peak, it's at an earlier time than the one we saw here, which is great because now we have people who are, you know, they're unfortunately still getting sick, but hopefully they're in that age group that can recover a little bit better from this infection. And also the peak is much lower. So we're not going over that healthcare capacity. If the older age groups get infected after this point, there will be plenty of resources and healthcare services available to them. So this is really great. And this is just one example where we release the young age group first, right? Anything could happen, and we can experiment with these different strategies to see what would work out the best. So this just gives an idea of what types of things we can do. All right, but wait a minute, we have a vaccine, right? So now we didn't at the time that this paper was drafted. So what would we need to do to change this model to something that would include a vaccine? Well, we'd probably add parameters that have something to do with the vaccination rate, you know, how many people are actually getting it, and doses, and maybe possible boosters down the line. What's the efficacy of the vaccine? That would be important as well, especially as we go and talk about herd immunity and that kind of thing, which I know Dr. Moran's going to talk about in a little bit. Maybe we have some sort of waning of immunity with this. And Can vaccinated people still get COVID? Yes, absolutely. So there's some sort of partial immunity going on too. We'd want to include all of these things. And right now, unfortunately, the vaccines are still very new. And so we don't really have a whole lot of information right now because everyone who's working on these types of models, they're still working on them and they are actively being published right now. But the CDC and the WHO, NIH, all of these organizations, they actually do use this type of modeling to help them when they're making public policy decisions. So we know that they're getting their information from lots of sources, but this is just one of those ways that they can see, how to proceed from there. And I'm going to pass it off now to Dr. Moran, who will talk more about, well, everything, but the vaccine in general.
All right. So thank you, Dr. Alvey. So when we're talking about vaccines, what we're really interested in is our adaptive immune response. And this is our natural response that we've evolved over time to deal with infectious agents like viruses. And so looking at this graph here, we're kind of simulating what happens with the immune system in response to a viral infection here. So at time 0, somebody gets infected, and it takes a little while, about 7 to 10 days, but eventually your adaptive immune system is going to kick in and it's going to start producing effector molecules like antibodies or cells like T cells that are going to be able to respond to that virus, and they're going to build a cloned army of cells that are going to fight really hard. That's why we often feel kind of lousy when we're dealing with an infection, because it takes a lot of energy to get those effector molecules and cells going. And in most cases, that ends up being a success. We clear the infectious agent and we're healthy again. But the real interesting thing here is that we've evolved this capacity for memory. And so in addition to these effector cells, we have these memory cells, and they're very long-lived. And they're going to be sitting and hanging out and circulating throughout the body, so that if we encounter that same pathogen a second time, they're going to be able to respond very vigorously, very quickly, and very effectively. And in most cases, they're able to clear the infectious agent without having any symptoms. And often the person doesn't even know that they're exposed. And if we have that secondary exposure, now we're just going to be boosting the number of those memory cells we have in our body. And they're going to provide us for really long-term protection against that particular pathogen. The idea here with vaccination is to kind of mimic what happens during a natural infection, to be able to build that immunological memory without having to go through all of the nasty consequences of having that first exposure and the risks associated with that initial infection. So again, we're going to be talking about these adaptive immune cells and some of the molecules they produce. So one cell is our T lymphocyte. The T lymphocyte is going to respond to a virus, like COVID, for example, and it's going to build this cloned army of cells. And so in that army, you have a number of cells with the E here. Those are our effector cells. And then you have a few of these cells, the M cells, which are our memory cells. Again, the idea of our effector cells is to go out and fight and do their thing to clear the infection. The memory cells are going to be those long-term cells that are going to hang around for quite a while. Our other main adaptive immune cell here is the B lymphocyte. And so our B cell is going to respond to a virus here. It's also going to build a cloned army, and it's going to generate effector cells here in yellow and memory cells here in the orange. B cells are a little bit different. Their effector cells will differentiate into these plasma cells. And the job of plasma cells is to make these molecules called antibodies. And then again, the job of the memory cells is to stay in circulation for many, many years and be available to respond if we see the pathogen again. So what do these antibodies do? Well, they're going to bind to these molecules on the outside of the virus. And this is what we call neutralization. And the real benefit is when these antibodies are going to block the ability, for example, of these spike proteins on the virus from being able to interact with the receptors and therefore block the infection. But we can't underestimate how important these T cells are. The job of the T cells is to go find those cells that are already infected by a virus. So for example, this might be the bronchial epithelium in somebody dealing with SARS CoV-2. The job of that T cell is to kill this cell here infected by a virus. And the reason we kill the cell is because this cell becomes a factory for producing lots of more virus, which can go and infect other cells. So the job of our T cells is to get rid of those infected cells. And when we think about developing a vaccine, well, the best vaccines are ones that elicit both of these effector and memory cells for both T cells and B cells. So when we are thinking about vaccines, historically, there have been a number of different strategies that researchers have used to be able to try to create a vaccine. So the first one is to have these killed or inactivated viruses. So because they've been killed or damaged, they're no longer able to cause an infection, but they are able to stimulate the immune system and generate those effector and memory cells. We also have live attenuated viral vaccines. So these are infectious viruses, but they've been modified in some way so that they have a reduced ability to grow and replicate in human cells, and they're no longer pathogenic. So they cause a limited type of infection, but the immune system is easily able to overwhelm these attenuated viruses and produce that immunity. We also have these slightly more recent subunit types of vaccines. And so in here, you're not using an entire virus at all. You're just using a piece of the virus. Very often, it's a little protein part of the spike protein that is injected into somebody. And that, again, is used to produce very often these neutralizing antibodies. Again, there's no virus, so this can't cause any kind of infection. And right now, the Novavax company is undergoing clinical trials for a COVID vaccine. It's not available yet, but this is one of the vaccines in the works. And so these are the three vaccines that we know a lot about. These are the ones that have been permitted to be used in the United States, although they're not completely FDA approved yet because they haven't gone all the way through phase 1, phase 2, and phase 3 clinical trials. They received permissions to be distributed because the initial clinical trials showed such positive results. And there was a desperate need to initiate vaccination of people. So the Pfizer, biotech, the Moderna, and the Johnson & Johnson's Janssen vaccine. So the first two, you get the 2 shots for the vaccine, and you're fully vaccinated 2 weeks after you get the second shot. For the Janssen vaccine, it's 1 shot, and you're, again, fully vaccinated after the 2 weeks after that 1 shot. And so what are these 3 vaccines? These are nucleic acid vaccines. And many of us, we've heard that these first 2, the Pfizer and the Moderna, are these mRNA vaccines. So here's what that means. Dr. Merkhofer talked about RNA as being the genetic code for some of these viruses. Here, we're not going to use the code for the whole viral genome. We're just going to use mRNA code for a piece of the spike protein, a little bit of the spike protein. That mRNA is going to be encased in a lipid coat, and that's going to help it enter our cells. Our cells are going to turn that RNA into viral proteins. So some of those spike proteins, they're going to display them on the surface of our cells. This is going to allow our immune system to respond to those viral spike proteins and get those B cells and T cells activated. So again, there is no full virus here. It's just a code for part of the viral spike protein. So you can't get COVID from these types of vaccines. The Janssen vaccine is similar in some ways and different in some other ways. So this is a DNA coding for the spike protein. And in this case, instead of embedding that DNA into a little lipid vesicle, it's going to be carried by a virus, another virus that infects humans. Dr. Merkhofer introduced the adenovirus as a virus that infects human cells. But this is a adenovirus that is not capable of replicating in human cells. So it's really just a delivery mechanism to get that genetic code for the coronavirus spike inside the cells. So once again, because the adenovirus can't replicate, can't cause disease, there's no risk associated with having any type of infection going on with this vaccine either. So the DNA codes for the spike protein. Once more, it gets expressed and our immune system gets triggered to make those antibodies and T cells. Are these vaccines effective? Well, the answer is they absolutely are. And there's many studies, not just the original clinical studies, but every single day we're seeing additional studies that are continuing to support the safety and efficacy of these vaccines. But here's one group that's interesting to look at. This is, the CDC has a number of trackers where it can look at different regions of the globe and different states within the United States and then different populations. So here we're looking at COVID-19 cases among nursing home residents. And the reason this is informative is because this group was among the first populations to receive the vaccines when they got rolled out. And so you can see here over time, you had the cases in nursing homes, and there was this really big second wave that was coming into the nursing homes in December and in through January. But it's at this time in January that the first vaccine started rolling out. And you see an immediate and very clear impact of vaccinating nursing home residents, and the number of cases has just absolutely dwindled in this case. And so when we think about trying to get everybody vaccinated, you can think about it, the CDC has this continuum that they talk about in the level of confidence that people have in receiving a particular vaccine. It doesn't have to be the COVID vaccine. But over on the left, you have people who are really just refusing. They're not going to get vaccinated. You have people who have this passive acceptance. And these are the folks that are saying, well, I'd like to wait and see what happens. I'm going to check and see what the other people do when they get vaccinated. And I want some more information. I want to know what's going on here. And then you have the people on the right who have completely embraced the vaccine. And that's when you have the maximal demand for receiving that vaccine. So what we would really like to do is to hopefully convert some of those passive acceptance, wait and see people into fully confident people, because there's a lot of evidence to suggest that's what's going to provide the best protection for our population. So how do we build that public confidence in vaccines, especially when we have this new technology, relatively new technology that's being used? Well, when you talk about these things, it's really important to stick to the facts and to utilize reputable sources. Historically, and I'm going to talk a little bit more in a moment, but there are a lot of sources of misinformation about vaccines, and those really feed into people's fears about whether they should get a vaccine or not. And so it's important as citizens here to really stick to the facts and make sure that we are talking about the vaccines using the science that's available. And what we want to do is emphasize the potential consequences of staying in that wait and see category. And so this is an example that you may have heard of in the United Kingdom. And this is not about COVID, this is about measles. And so what we're looking at here is over time, so starting in about the early 90s, going into the early 2000s, in blue, the outbreaks of measles. So the measles vaccine was around in the 1960s, but in around the late 1990s, parents were required to get their children vaccinated with the MMR, measles, mumps, and rubella to go to school. And so you had a lot of people getting vaccinated, and that was pretty effective at controlling measles outbreaks in the UK. But in the late 1990s, there was some misinformation, both in the scientific literature as well as being promoted by some prominent people in the media, that started this anti-vaxxer movement for the most part, and encouraged people to be afraid of vaccines because perhaps there was this relationship with autism and the vaccine. This has been completely proven to be false and discredited. The folks who published that information have had their articles removed from circulation. That's really hard to even find these days. But the consequences are people started dying. Whole communities in the United Kingdom started to not vaccinate their kids, and this is what happened. We've heard a lot of this term herd immunity being thrown around. And in the media, they talk about, well, as soon as we get to herd immunity, that's what we really need, because that's when life can go back to normal. But we have to be really careful with this term, because herd immunity is not something that we just hit it and then we're fine. Herd immunity actually takes constant maintenance for it to be sustained. The idea here is you don't have to vaccinate everybody in the population. As long as you get most of them, the virus will have nowhere to go. But in today's world, where we don't have geographic isolation, we have people traveling, and we have people's behaviors that can change quite frequently with the media, just achieving herd immunity once is not going to be enough. We really have to encourage everybody to get vaccinated if we want to show protection. All right, so Dr. Merkhofer talked about these variants. Are the vaccines going to protect against the variants? That's a question. And it's a concerning one, because there have been some scientific studies released that suggest that, well, some of these variants might not be responding very well to some of the antibodies that are produced in response to COVID. And so this study is looking at antibodies from convalescent serum. That means from people who have gotten COVID and recovered and have antibodies in their blood, as well as those that have gotten vaccinated to generate those antibodies. And it saw that some of those variants were not neutralized quite as well with these antibodies. But they're also very quick to talk about the limitations of those studies. These studies are typically done in the Petri dish. That's what in vitro means. And they're only looking at a virus's ability to infect a cell in a Petri dish. And the problem is that doesn't take into account any of the other components of the immune system, other than simply an antibody's ability to block a spike protein and a receptor and having that virus get into the cell. And the truth is, there's a lot more to the immune response than just those neutralizing antibodies. In particular, we have the T cells. And so the T cells are what's going to save the day here. So this is a very recent study. It hasn't even gone through peer review yet. It's a preprint, but these folks will get this published very soon, showing that the T cell response to these variants is extremely encouraging. When looking at the wild type, or the first iteration of COVID, SARS, that came out, and now some of these variants, we're seeing roughly equal T cell responses to all of those different types of virus. And so this is extremely encouraging that the T cells are there. So kind of to wrap up, all of the data supporting vaccines has been really positive thus far. The clinical trials are showing great safety and efficacy, protecting people from severe COVID and death, as Dr. Merkhofer pointed out. And really, this new data with the T cell response with vaccination is likely going to be able to pick up any slack that's caused by perhaps some decreased activity of neutralizing antibodies with these variants. And there's a lot of reasons for that, and we can talk about that if you're interested with the questions. But another facet here that's really important is that these vaccines, this new technology using nucleic acids, well, they are much easier to modify and mass produce than some of the more earlier traditional vaccines. And that means we can move on the fly. We can develop a new vaccine very quickly if needed in response to these variants and get it distributed out to people. So our response time is going to be much improved. So there's a lot of reason to be very hopeful and very positive about the efficacy that these vaccines are going to have. And with that, I think we will turn it over for questions. So thank you all for listening, and we'd be happy to do our best to see if we can answer some of the questions you have.
That was great. Big round of applause for you all. So questions? Folks can feel free to raise their hand. And Vince, you want to start?
One of the reasons I basically held out for the Johnson & Johnson Janssen variant is because I have, now this is just personally me, nothing that I've researched very much, but my thinking is that the way they've created the virus of the antibodies and stuff with the DNA version has been around and tried and true for decades. Whereas the mRNA is kind of a new technology that I haven't quite embraced yet. But after seeing this presentation today, it's making me think a little bit more about it. But that's why I stuck out for the one shot.
Yeah, and that's completely understandable, right? This new technology is something that people haven't heard a lot about before. But I think one of the keys here is, what do we mean by new technology? So mRNA vaccines have been around for quite a while and been tested in animals for quite a while. And there have been even some applications in humans for mRNA vaccines in some severe cases of cancer, where the vaccine's trying to boost the immune system to cancer as well. So although this is really the first time that it's been used on a very large scale in human populations, the technology has been around for quite a long time and is something that has been pretty well vetted in those animal models. But yeah, you're absolutely right. There has been reason to question anything that's new out there. And I think we look at the data again, and the data is very promising about the efficacy and safety of this moving forward.
And that's what I took away from the presentation. But my basic apprehension goes back to that slide, was in vitro versus in vivo. The DNA has been around a lot longer than the RMNA. So.
Well, I mean, to be honest, though, the Johnson & Johnson version of the vaccine is brand new as well. That has never been done with, it's a chimpanzee-based adenovirus.
Yes.
And really, these adenovirus-based vectors have really only been used for gene therapy in the past. They haven't been used for vaccines. So in essence, all three of these vaccines are new in their use for delivering vaccines. But that said, the trials have been the trials were of quite a good size for how quickly they were able to put them together, and the data was pretty overwhelming about the safety. And a lot of people do ask, what's going to happen 6 months down the line? What's going to happen 12 months down the line? And of course, we can't predict the future, but at the same time, there's no evidence based upon previous types of vaccinations and what we know about this method of vaccination that any sort of reaction would occur outside of 4 to 8 weeks or so. And the data really does support that.
Yeah, I agree. I mean, it's just one of those things, as I said, like, it's just a personal feeling of mine that the old tried and true DNA modification of gene splicing is a lot more than the new RMNA. That's all.
No. So there's a question in the chat. It says, Dr. Moran, could the type and severity of symptomatic response to COVID vaccine be influenced by a person's previous exposure to COVID?
Yeah, that's a very interesting question here. And, you know, I think Dr. Merkhofer just said something really relevant here. Timing, when you look at the response that people have to the vaccine, is really informative here. So there's kind of two different responses that people have when they're vaccinated. One is kind of this immediate response, and that's often more associated with hypersensitivity or allergy. So a lot of times if you have something, that's why they sit us down for that 15 minutes after we get the vaccine, looking for something that happens right away and could be potentially severe if you are allergic to some component in the vaccine. And that, it turns out, is pretty rare. But your question about if somebody had COVID infection and recovered and then got the vaccine, would they experience something different symptomatically than perhaps somebody who had not been exposed to COVID? And I think the answer is you may have a little bit more of a robust immune response there, because essentially then the vaccine is kind of like a booster shot in some way. And so just like for those of you who got the 2 doses of the mRNA vaccines, very often we felt a little bit, you know, perhaps just a little bit of soreness after the first one. But I know myself with the Pfizer one, after getting that second shot, I felt like I had an elephant under my armpit there. And that meant my T cells and B cells were responding very vigorously, building a cloned army in my lymph node. And I felt a little bit lousy, because anytime your immune system gets going, it takes energy away from other things. And that's why we feel a little yucky. So there might be some difference for somebody who's been through COVID when they get that vaccine. But overall, it's going to be self-limiting because it's not an active infection, and it's going to get better quickly, and then you're going to be protected.
So I'm going to take a question right here. What should we expect for the future of mRNA vaccine? Should we expect common vaccines like the annual flu vaccine to be offered as an mRNA vaccine in the near future? I think it's a really good question. I think we are going to see mRNA vaccines to be used at a much higher level than they are right now. Influenza is a tricky one, and influenza is a tricky one because there are, when most influenza vaccines are what are called either trivalent or quadrivalent vaccines, where there are 3 or 4 strains of influenza that you're getting vaccinated against. And we're not talking about variants here, we're talking about actual completely different types of viruses. So it would be a little more difficult to do an mRNA vaccine against the flu because of all the different components you would have to include. Because when you do get, for every strain of that influenza vaccine, there is both an HA protein and an N protein. So the number of parts of the virus that would have to be made in the cells starts to increase dramatically. But I think one place you are going to see mRNA vaccines a lot more, and kind of Dr. Moran alluded to this a little bit, is actually going to be for things like cancer. Some of these diseases where we have had trouble targeting specific therapies, I think we are going to see this technology to be used. It wouldn't necessarily be a vaccine for cancer, but I think you will see the mRNA technology being used for something like cancer.
Could I jump in here for a second? Because this is a question I wondered about. What about a vaccine for HIV?
So vaccines for HIV, and Dr. Moran can speak to this too, are very, very difficult. And part of that has to do with the mutation rate for HIV. There is much higher. And maybe Dr. Moran, he's a seasoned immunologist. He can jump to this as well.
Yeah, as Dr. Merkhofer said, HIV's rate of mutation is very, very high. Right now, the idea with COVID, it appears that the mutation rate is significantly slower. But aside from that, the real problem with HIV here is the fact that the virus infects and kills the very cells of your immune system that would be responsible for clearing the infection. And so while you're trying to do everything you can to boost the immune response, the virus goes and changes, and then you start over again. So in HIV, it's this race between your body's ability to produce new immune cells every time the virus mutates, and the virus wiping out those immune cells as they infect them. And it's a war of attrition, right? It's one that eventually, without any kind of treatment, that people lose that war. But in the case of, for example, Magic Johnson, who is able to afford all of the fancy designer antivirals that are reformulated every time the virus mutates, they're kind of at that steady state where the race is still on. And fortunately for folks, you may have started to see some of those commercials on TV for some of those antiviral therapies for HIV. So they're becoming more and more accessible. But vaccination for HIV is going to be a problem.
Great, thank you.
So there's a couple questions here for Dr. Alvey. One question, did we use a mathematical model like the one you described for making decisions on campus? And also, given the fact that it appears to be that at least in a handful of states, there is an increase in the number of cases right now, do you think that the infectious rates could start to look like the graph you presented, in which the younger individuals are released first?
Okay, so to answer I'll go in order here. The first question about using models like this to make decisions on campus. No, and the reason is the data doesn't really exist for such a small population as our campus at the moment. Health services has to be careful with what information they release to me or the public. It's a lot of sensitivity issues. So using a model that would be more broad for New York would give different results than one for a college campus, and we'd have to be mindful of that. As far as infection rates and possible shutdowns in the future, or if we're really going to start looking like that staggered release graph, it's possible. The thing is, though, and I see the question talking about spring break and traveling and that kind of a thing, not everyone's doing what they should be as far as wearing a mask and social distancing. And we have to make assumptions when we create these models that at least to some extent, people are doing that. So all of the results that we are getting and all of the parameter values that we choose to use in these models are taking that into account. And so I'm not sure we're going to get an exact, replica of that graph, but yeah, definitely people are starting to go back to work in person. People are going to school on campuses. To some extent, we're going to see more cases in that younger group and hopefully less in the older group. And that's a good goal. But also keep in mind, we did not have vaccine in that model. And that is a very, very important part of the picture right now.
So I did answer a quick question in the chat. What if you didn't have much of a reaction after receiving the vaccine? Does that indicate your immune system isn't as strong? There's no evidence to suggest that the reaction to the vaccine is correlated in any way to your immune system's ability to fight off the infection. There are differences due to age. There are differences due to genetic background. There's no evidence that. So if you got your second shot, you didn't feel anything, don't go out and get a third shot. Not yet. Now, there is a question here that actually came up from another person before the talk. So asking about the rumors of fertility issues when it comes to vaccines. Is there any real evidence to support this? Did you want to take this one, Dr. Moran?
So to be honest, I'm not very up on social media. So this is the first time I'm actually hearing about this. My feeling is that, again, in the scientific literature, there's been nothing to suggest that there's a problem with cross-reactivity with, the viral spike proteins and human proteins that are going to put somebody at risk for having miscarriages. So, again, this is one of those things where we have to be very careful about the potential for misinformation getting spread, right? This is what's going to contribute to that wait and see population here. When we have kind of stories like this that show up in the media without being validated by scientific evidence types of things. Yes, is there, perhaps a scenario in biology where we've talked about molecular mimicry, where maybe a pathogen has a molecule that's similar to something in the human body. That is certainly something that is happened in other cases, but there's no evidence that this is truly something that we've seen with corona.
Yeah, and I'd follow up with that to say, while there is no evidence that there is any issue with fertility issues, I think whenever you're making a decision like this, it's always a bit of a cost benefit analysis. I think the cost of getting infected with SARS-CoV-2 is going to outweigh any anecdotal evidence that people seem to be reading about when it's not been supported by anything that's been presented data-wise. So someone asked, how well conserved is the ACE2 receptor? For instance, why were there not as many house pets getting sick from the virus? So it's a good question. Viruses are what are called tropic. So they only have the ability to infect a very small range of organisms. It can be within only some species. Some types of viruses that infect bacteria can be up at the genus level, but generally they're very, very specific. And that has everything to do with the three-dimensional structure of the receptor that the virus binds to. So if you looked at the ACE2 receptor between canines and humans, you will probably find some similarities. But the reality is that even the smallest difference can really have a dramatic effect on the three-dimensional structure of the protein. And the way that the viruses interact with their receptor has to be exact in order for that type of infection to occur, in order for adhesion to occur. So there are some viruses that can go across species, but most of them are very specific. So Chris asked, can you touch on the ability of the virus to reproduce and be spread to an unvaccinated person from a vaccinated person? So I think this is a big question that everyone has right now. The CDC just came out with some very nice data about 5 days ago that addresses this. So the clinical trials were not designed to look at this question. So that is one thing everyone, and this comes up on social media. I'm gonna get Dr. Moran a Twitter account so he can follow all this. But this is one thing that comes up on social media a lot, is people who are arguing against the vaccine say, well, there's no evidence that it actually prevents the spread of infection, because the clinical trials were designed to look at symptomatic infections and not asymptomatic infections. But the CDC has now a lot of data that just got released 5 days ago that shows that they only looked at the Pfizer and the Moderna. There's no reason to think that Johnson & Johnson would be any different, that those vaccines protect against asymptomatic spread at a rate of 90%, which means that if you are vaccinated, the chance of you getting infected and spreading that to an unvaccinated person or another vaccinated person is 10% or lower. It's a very small amount. So it really has to do with how much virus can your body make. If you're vaccinated, it doesn't mean that the virus gets to your mouth and then bounces off. What it means is that the amount of virus that you're exposed to is not going to be able to replicate to a point in your cells that you will produce enough virus to expose someone else that will make them sick, particularly if they're unvaccinated. And if they're vaccinated, well, then you have the same thing going down the line. So the new data is really compelling. That suggests that vaccinated people are not going to spread this. Now, could variants be different when it comes to this? It's possible. It's hard to look at that type of data and break it out by variant, because the lack of infection, when you're measuring the lack of infection, you don't know what type of strain that person who didn't get infected was exposed to, because there wasn't an infection. But as of right now, the data suggests that it's not likely to happen.
Any other questions for our presenters? If not, I really want to thank you all very much. This has been highly informative, and of course is the topic that is top of mind for all of us, I'm sure. So the opportunity to hear some clear, scientifically based information is very refreshing, considering what we're all exposed to, everybody but James, on social media. And to kind of debunk some of these fears, which, as I was sharing with the folks before we got started, that I have heard these very same claims from staff members at the college. So this kind of misinformation can really affect people who you would normally think would be making solid decisions. So let's hope that we can all go out, at least from this talk, and share some really good, solid information.